Hi everyone, and welcome to today's Daily Booster. Now, today is number 6 of our biology boosters. What we're going to be looking at in today's booster is photosynthesis. So first thing we really need to know then is that when we're talking about photosynthetic organisms, these are the ones that are using sunlight in order to produce their own food. So what we can actually say is that these photosynthetic organisms are the main producers of food and, as a result, biomass for all life on Earth. One of their favourite multiple choice questions here is asking you about the type of reaction that photosynthesis is, and it is an endothermic reaction because it's taking in that light energy in order to actually carry out the reaction. The little diagram on the right is just giving you a few reminders about the fact that once we've made the glucose, it can be used in a whole range of different things. We can store it as either starch or sucrose. We can convert it into cellulose to make the cell walls. We can even change it into things like proteins or fats and oils for other purposes within the plant. As far as photosynthesis goes, we need two chemicals at the start: carbon dioxide and water. Carbon dioxide comes from the air around the plants, and it will enter them through the stomata in a process of diffusion. Now, diffusion is going to come up in B2, but just as a reminder, that's where those chemicals are moving from an area of high concentration to an area of low concentration. The second one, water, that comes from the soil, and it'll enter the plants through their roots by the process of osmosis. Again, we'll look at osmosis in more detail in B2. Once we actually have the water and the carbon dioxide inside the plant, we need the light energy, and this comes from the sun. As the sun shines on our plant, what happens is we're going to trap that light energy by using the little subcellular structures called chloroplasts. Now, the chloroplasts contain this green pigment called chlorophyll, and that's what traps the light energy. As far as photosynthesis goes, you need to know that it is a two-stage process. So we have stage 1, the light-dependent stage, the one that needs the light energy, and that energy is used to split the water molecules into oxygen and hydrogen. So water, H2O, what we're doing is we're splitting the oxygen and the hydrogen from one another. Then stage 2, the light-independent reaction. This is where the carbon dioxide then combines with those hydrogen ions to make the glucose. What we then come on to are a range of different experiments that we can carry out to investigate photosynthesis. And the first part here is looking at how we actually carry out a test for starch. Now, we touched on that in one of our previous boosters where we were looking at the food tests. So to test for starch, we use iodine, and it goes from orange-brown to a blue-black colour. Now, the way that we do this with our plants is we take a leaf, put it in a beaker of boiling water for a minute just to kill it, to stop any reactions occurring. We then place it into a boiling tube containing ethanol, and then we're going to heat that for 5 minutes in a beaker of boiling water, not over a Bunsen flamer. The reason we put it in the ethanol is it removes the chlorophyll from the leaf, and the reason we don't use a Bunsen flame is because ethanol is flammable. So you literally have a beaker of boiled water that you then just sit it in. No flames anywhere near because we don't want to set fire to it. Once that's happened, we take it out, rinse off the leaf, spread it on a white tile, and then we add the few drops of the iodine solution. If it goes blue-black, starch was present. No blue-black, starch was not present. Do note the point at the bottom that before we carry out these photosynthesis experiments, the first thing we need to do is de-starch the plant. Now, by de-starching the plant, this is a very high-tech method. You stick the plant in a dark cupboard and leave it there for 24 hours. So, let's look at the individual experiments that we could do. The first one, if we needed to prove that chlorophyll is needed for photosynthesis, we use a type of plant called a variegated plant. So you can see its name there, variegated. All that means is it's got the green areas and the white areas. So if you look at the little diagram on the right, you can see there are green bits and there are white bits. Green bits have chlorophyll, white bits do not. So after we've destarched our variegated plant, we then take it out of the dark cupboard, stick it in sunlight for a few hours, and then we test the leaf for starch. What you'll find is that green bits will have starch, the white bits will not have starch. Experiment 2, to prove that we need light in order to carry out photosynthesis. Destarch the plant first of all, but then what we're going to do is stick a bit of foil or card over part of the leaf, as you can see in the diagram on the right. You then put the plant in sunlight for several hours again, and then we're going to remove the card or the foil and then test that leaf for starch. What you'll find is that the bit that was covered by the card or the foil, no starch present. The bits that were not covered by the card or the foil have starch present. Experiment 3 is proving that we need carbon dioxide. So destarch the plant first of all, and then we're going to cover it with a polythene bag, and inside the polythene bag is a little pot of a chemical called soda lime. Now, soda lime absorbs carbon dioxide from the air. So all we've done by sticking it in the bag and giving it the little pot of soda lime is we're removing the carbon dioxide from the air around the plant. Leave it in sunlight for several hours, test the leaf for starch again. The last one, to prove that oxygen is given off, is a different experiment. It's not very easy to catch oxygen from plants that grow on land. So instead we use aquatic plants, we use pondweed. Now what we do here is we put our little bit of pondweed in a beaker with water. You put a funnel over the top of it, because that just means the bubbles are going to be directed to a narrow column. And then we've got a test tube upturned over the top. So the test tube is filled with water, and then what happens as the gas is made, it collects in the tube, pushing the water out. We can then remove the tube and then test it for oxygen. A glowing splint relights. Assuming that happens, we have oxygen. If it doesn't, we clearly have no oxygen. Now, when it comes to doing these investigations, the most likely one they will ask you about is using pondweed. What they're going to ask you is to do with the collecting gas or the counting the bubbles. So the simple method is you literally count bubbles, but this has problems. First of all, the bubbles are not all the same volume, so you won't necessarily have an accurate measure of the amount of gas made, just the number of bubbles produced. Plus, dead easy to miss a bubble when they're coming at quite a rapid rate. So we can upgrade that by collecting the gas in a measuring cylinder. So just like the setup we had to collect the oxygen, but this time we'd use a measuring cylinder instead of a test tube, because then you can just read the volume off the actual side. If you want to go one step further, then you could even say use a gas syringe at this point. Now, in terms of changing the factors that could affect the rate of photosynthesis for our pondweed, if you were to change light intensity, you would put your desk lamp close to the plant first of all, and then you'd move it further and further away, taking readings at each new distance. On those ones, they will sometimes have like a big measuring cylinder full of water between the lamp and the pondweed. And that's purely to absorb any of the heat from the light so that it doesn't also change the temperature. Option 2, to change the carbon dioxide concentration, we add different masses of potassium hydrogen carbonate to the water. Or option 3, to investigate temperature, we just carry out the experiment in a water bath, which we could then set to different temperatures each time. Final thing for today then is to have a little go at the quiz to check how well we understood it. So head on over to have a go at the little quiz, and don't forget to come back same time tomorrow for another booster.